About our experience on opening up human subject research safely in the wake of the COVID-19 pandemic. You can see myself here as well as my other co-conspirators in this presentation. So the University of Nebraska Medical Center is located in Omaha, Nebraska. We are a 718 bed tertiary care center that is primarily an academic partner with UNMC and Nebraska Medicine. It's the largest hospital in the state. Nebraska Medicine with UNMC through its Center for Global Health Securities are leaders in the state pandemic response. We also work in collaboration with the Children's Medical Center that's also in Omaha, Nebraska, focusing on healthcare for children. UNMC also sponsors the public health laboratory that led the SARS-CoV-2 testing for the state. So a little bit about our COVID-related timeline. So University of Nebraska Medical Center was kind of in the spotlight, a national spotlight back in February of 2020 when UNMC accepted persons from the Princess Cruise, as well as those individuals who resided in Wuhan, China, in our national quarantine center. At that same time, the University of Nebraska joined NIAID in their sponsored startup trial with remdesivir for patients with COVID-19 infection. Across the country and across the world in March of 2020, we all know and can remember back that COVID-19 case counts were beginning to increase across certain countries, but started to see that it was going to spread likely across the world. In April of 2020, our university implemented pandemic plan restricting essential workers only. At the same time, we started having discussions with our IRB on how to restrict or pause human subjects research involving face-to-face contact to only allowing COVID-19 trials and other trials with significant direct benefit to continue to proceed forward. In May of 2020, our cases initially started to decrease, and we began discussions on how to reopen or unpause human subjects research again. So a little bit about our how we evaluate human subject research under our own institutional IRB. Our IRB has robust human subject research portfolio, handling investigator-initiated trials, federally funded trials, and commercially sponsored clinical trials, both in first-in-human phase 1s all the way to post-approval, post-marketing phase 4 trials. This does include many high-risk patient populations, including patients who have cancer, solid organ transplant, complex congestive heart failure, special programs in cystic fibrosis, neuroscience, as well as on the imaging modality with neuroimaging. We also, our IRB will cover our Children's Medical Center protocols, as well as other community-based protocols focusing on rural communities, immigrant populations, nursing homes, meatpacking plants, and other farm-related research given the unique geography of Nebraska. We're also the sole IRB for our Buffett Cancer Center, again, the Children's Medical Center, but also our Monroe Meyer Institute for Genetic Diseases and Rehabilitation, and also work with a partner university, the University of Nebraska at Omaha, in regards to many protocols going through psychology, biomechanics, and exercise research. So very broad spectrum of clinical trials that are being seen at our IRB. It also can serve as a single IRB for certain research consortium. So kind of along with the pandemic, we started having discussions on when and how to open up research after we had initially paused in regards to the rising caseload, but also the concerns for the impact on personal protective equipment supplies, namely surgical masks, as well as N95 masks, and really felt that it was important to prioritize these resources for clinical care. And really there was uncertainty and still some uncertainty about PPE availability, both for the clinical standpoint, but also how is this going to be allocated from a research standpoint. We also factored in risks to human subjects. As we know, early on, there was suggestions of who was going to be the highest risk populations. typically and falling out, often those individuals of advanced age, but really focusing on the research itself and what was the patient population for human subjects that were potentially going to be enrolled. We also considered a risk to research personnel as well. And what was going to be the impact or the burden to the clinical facilities and to the staff. So acknowledging that not only do human subject research interact with the clinical staff, but they may also have research required studies like radiology required studies, and also pulmonary function tests, which would bring them into contact with our clinical realm, but also potentially higher risk situations like PFTs. We also recognize that this was a rapidly evolving space where there was likely to be not only institutional new policies, but potentially also state and federal policies and procedures as COVID-19 became more apparent and surged in different geographical locations across the country. We also acknowledge that there could be new biosafety knowledge that would come out of COVID-19, and research personnel needed to be aware of this, and noted how this could impact their protocols if they were to need to restart, depending upon what was being collected at the time. And then we needed to and wanted to meet certain contract considerations, meeting deliverables, both in startup timelines of studies, but also studies have amendments going through, and not wanting to lose valuable research information, and including discussions on whether or not to allow study monitors on site as stay-home orders were in place, but also to make a transition to finding out how we could do things that didn't require face-to-face monitoring, or otherwise later known as virtual monitoring. So we took a general approach to reopening human subject research safely. Like many centers, we opened research in stages. There was a phase 0 stage, and so this was research that was never paused or never stopped. This included our COVID-related research trials, as well as other trials that have life-saving therapies embedded within the research, which included our cancer, some of our cancer trials, as well as trials in cardiology, for instance, acute myocardial infarction trials. Phase 1 was deemed as direct benefit being conducted concurrent with clinical care. This also included data registries and biobanking protocols conducted concurrently with clinical care. And then our phase 2 was remaining protocols with direct benefit research not concurrent with clinical care. And then phase 3 as studies with no direct benefit. So perhaps not like other centers, we required our principal investigators to submit a safety plan in response to the COVID-19 pandemic. And certain elements were required as part of that, or answers from the PIs and the research team on how they were going to handle restarting their research amidst the pandemic. That included implementing or continuing certain remote processes where possible in their research, how they will, answers on how they will screen research subjects that are coming into their facility. And again, we had a broad representation in our human subject research, all the way from at the University of Nebraska at Omaha, doing biomechanical research, all the way to therapeutic trials, occurring in our oncology building. We also asked them to show support that the director of the unit welcomes the study back. We were concerned about resources, chair times for certain populations, as we had put kind of a stay-at-home effect, and then patients were also coming back into the clinic to restart therapies or restart interventions, radiology, and PFTs, and we didn't want to overwhelm the system. And so letting the directors of the units know that research was restarting and making sure that they were okay and felt the volume was going to be appropriate. We also asked them to describe the plan for the cleaning protocol, not only in the clinical space, but outside in the research space. That includes, functional MRIs, treadmill cleaning protocols, for example. And then we asked them to describe the samples that were going to be taken to assess for any new biosafety strategies during the collection or processing. So this included saliva testing, nasal swabs unrelated to COVID trials, and space for sample processing. So again, kind of a new environment and a new thought process had to be taken into place given the COVID-19 pandemic. And then also awareness and the ability to cover required costs of SARS-CoV-2 testing by that center or that setting. And so if there was a research non-billable PFT in place that wasn't going to be billed as standard of care, we engaged the investigators on how the PFT's testing for SARS-CoV-2 was going to be performed from a research standpoint. And so this safety plan was reviewed by a separate committee called our Human Subject Review Safety Review Committee. So a long, another acronym that was put in place and stood up by Dr. Larson with several members from our research community. And this was assessed before the IRB conducted their final review to allow for an unpausing of research. So again, kind of here are our timelines. So we opened up research. Again, our phase 0 never stopped accruing. Our phase 1s opened as COVID-19 cases were declining in May of 2020. Phase 2 opened 1 month later in June of 2020, and phase 3 again opened up in June of 2020, but required our safety plan review by the committee I previously mentioned. So in summary, our safety review committee was well received and quickly integrated into the research process. The cleaning and biosafety standard operating procedures were developed very quickly and with some ingenuity. The conversion to remote process were expanded to allow potentially consenting electronically through our own homebred research system. The committee reviewed occurred within 1 week of form completion. And so now we've reviewed over 200 research protocols for restarting. Funded studies were reviewed, were prioritized over unfunded studies. As part of this process, we uncovered certain knowledge gaps around clinical policies, as well as biosafety risks within certain protocols. It also, I believe, allowed for a decrease in anxiety on the nursing administration, that they were going to be flooded by research subjects and that would overburden the nursing staff as they kind of worked their way through the pandemic and patients, coming out of the stay at home kind of mantra that dominated the spring and early summer. And it also increased research administration awareness of just how many studies were being conducted and where. And now the committee continues to expand, and we handle on-site monitor visits, although we continue to encourage remote monitoring. So they have to request an on-site monitor with certain caveats to ensure that this can be done as safe as possible. And with that, I will conclude my talk. Thank you.
Thank you. And with no further ado, I will next introduce Dr. Melanie Cushion, the Senior Associate Dean for Research, University of Cincinnati College of Medicine, Senior Research Career Scientist, VA Medical Center, who's going to begin to share her slides and speak to the rapid research response to the COVID-19 pandemic at the University of Cincinnati. Melanie.
Okay, thank you so much. Let's see. All right, how's that look? Okay,
Great.
Perfect. All right, good morning to all of you, and thank you very much for the invite. As was said, my name is Melanie Cushion. I'm the Senior Associate Dean for Research here at the College of Medicine, which is part of a medical center complex that has both the undergraduate, graduate, children's hospital, and then our adult hospital as well. I'm honored to present the rapid research response to the COVID-19 pandemic by the university. I'm presenting on behalf of researchers, faculty, staff, and administrators who all came together to make these efforts possible. Okay, great. So shown on this slide is the timeline for the research lab response to the COVID-19 mandatory shutdown and re-entry at UC. On March 10th of this year, the College of Medicine was notified that all research labs were to submit disaster plans that would outline the process of shutting down the labs until further notice. These plans needed to include such things as cryopreservation of cell lines, the shutdown of sensitive instrumentation, and the like. Over 200 plans were reviewed and approved. On March 17th, the university and college began the transition to remote work with only critical personnel to remain on campus. At that time, we designed and issued forms to request critical research operations by the laboratories. The approval process focused on medical research that, if discontinued, would endanger lives of patients, operation of core facilities that were necessary to support critical research activities, ongoing animal studies, cryogenic maintenance, and COVID-related studies, etc., including names of personnel and the spaces that were to be affected and who would house the critical personnel and lab operations. Here we show the timeline for the RFA, the response, review, and award of the COVID-19 projects that we initiated at the very same time. Understanding that a rapid research response would be critical for our fundamental understanding of SARS-CoV-2, which there was little information about, and COVID-19, the sequelae or the clinical presentation. The Office of Research in the College of Medicine quickly partnered with the Vice President for Research at the university to fund innovative pilot projects focusing on SARS-CoV-2. A repurposed RFA, so it was recycled for pilot projects in general, was launched on March 17th. The applications were due on the 25th. That's quite a turnaround. Then the 29 applications that were received were reviewed with notices of award to recipients by April 8th. 6 faculty and Office of Research members reviewed all 29 applications, met as a review session, and ranked them. A total of $425,000 was deployed between the university and the College of Medicine to fund the pilots. Budgets were to justify 1 to 3 modules of $25,000 each, with benchmarking goals to be reached prior to funding of the next module. Shown in the slide is the clinical research response to COVID-19. So concomitant with the COVID project timeline I just showed you, the clinical research arm of the college, under the guidance of the Senior Associate Dean for Clinical Research, the Office of Clinical Trials, and a myriad of investigators and clinicians mobilized to establish a gateway for approving clinical trials, which is called the Innovation Committee, the collection of patient samples to create a biorepository and operationalize the process. This was the Innovation Committee and then a formed Operations Subcommittee. By July 27th, 114 patients had submitted specimens for the biorepository. And then by April, the first 3 clinical trials were approved. And by August, the pre-screening for the vaccine trial participants was initiated with significant URM populations to be enrolled. We are an urban campus and we are able to provide the NIH and the vaccine trials with a very diverse population of volunteers. Shown here is an update of the Innovation and Operations Committee. Over 100 potential studies were presented to the Innovation Committee that were related to COVID-19. Through the Innovation Committee and the Ops Subcommittee, a little less than a third were selected to move forward. And the total patients engaged in research is at 853, which includes those that submitted to the biorepository. And of those, 386 patients are in actual interventional and clinical trials. I'm just going to tell you a little bit about the progress that has been made, which has been significant. Here we highlight some of the projects, which include establishment of screening assays, a phase 2 trial, several manuscripts that are published or under review, T-cell experimental therapy, and field trials of homemade masks as to which fabric is the best. Our investigators who are making these discoveries are listed to the left, and really want to recognize them for the outstanding work that they are doing. So this is my silver lining slide. So there were some positive aspects that came out of all this madness. Our labs now have disaster plans that can be adapted for future disasters, which I anticipate will happen. Our institution initiated a gratitude campaign that recognizes the acts of kindness that our staff, students, and faculty have done for each other. It's amazing what human nature does under these times of stress. We have a more plugged in community with one another and also with our health system, who the research lab supplied with PPE during those initial phases and the shortages of the PPE, and now with the -80 freezers more recently. Our faculty and staff are more cognizant of safety, including the necessity for hand washing, not coming to work sick, not being that martyr that was sort of part of our culture, and better surface cleaning regimens at all. So we're much tidier and much neater and more sanitized. And in closing, we conclude institutions who can pivot and use existing resources can adjust to extreme circumstances, but we all need an all hands on deck approach. And I thank you for your attention.
Thank you, Dr. Cushion.
Alrighty.
Really appreciate it. And I'm going to then ask for Sophia Molina Garcia, Dr. Molina Garcia, Research Assistant at the UT MD Anderson Cancer Center, to begin to share her slides. And she's going to speak to the knowledge about COVID-19 among healthcare workers in Mexico, results from a national survey. Dr. Molina Garcia, go ahead.
Thank you so much for the introduction.
Yeah, your slides are not up. They were, and then there you go.
Great.
And you want to put that in presentation mode?
Yeah.
Okay.
Can you see it now? Thank you for the introduction. I'm Dr. Sofia Molina. I worked at MD Anderson, but I'm a physician from Mexico. And with other workers in Mexico, we decided to conduct a survey, a national survey, so that we could assess the healthcare worker opinions and knowledge about COVID-19 after looking at other surveys from around the world. So I'm going to present those results now. So if you don't know, in Mexico, we've had a huge problem with COVID-19, just as much as every other part of the world. But we've had more than 1 million infected, almost 100,000 deaths, and we've had more than 140,000 healthcare workers infected during the pandemic. Almost 2,000 of them died during this period of time. And you can see in that map of Mexico that almost all states in Mexico are between the red and the orange zone, which is the high risk, the high infection risk in the country. We decided to conduct this survey that was a free access survey for healthcare workers. It was designed on an online software. We had 16 questions, and they were around transmission, prevention, treatment, and the information resources that these workers used to get to know more about COVID-19. It was spread through word of mouth between physicians and other healthcare workers, and social media. We mainly used WhatsApp and Facebook to spread the link. And it was open from August 8th to September the 6th. We then used data for the analysis of the answers. We had a total of 516 responses. And as you can see in this little pie chart, most of them were from attending physicians. So 161 attending physicians answered the survey, 134 residents, and 87 medical students. But we also wanted to know what other medical staff knew about the pandemic. So we had 83 nurses, 32 janitors and other hospital staff, and then we also had lab technicians and radiologists answer. Out of all of the participants in our survey, we divided the country into 3 sections. So we had the north part of the country, which is in red, and then the center, which is in yellow, and the south. Because we work in the north part of the country in Monterrey, most of our responses were from the north. So we had 372 responses from the north. We also asked them about their main work areas. So 25% of the total answers said that they worked in the inpatient setting versus the outpatient setting. and 17% of those worked in the emergency department specifically. Some of the difference we saw between healthcare workers were that nurses reported more work in the ICU than residents and attendings. Residents reported more work in the emergency department, and attendings had more work in the outpatient department. We wanted to know where they were getting most of their information. We do have daily briefings on TV for the pandemic in Mexico, but we wanted to see if they were also getting their information from new published articles or social media. So we can see that attendings residents mostly answered that they were getting their newer information from peer-reviewed publications. But we had a large number of people answer that they had information from social media, especially the janitors and other hospital staff. We asked them if they had had any training, either online or in person, in their work areas. And nurses, 25% of nurses said that they did have a training. And students had some online training during the pandemic. So 29% of students said they had training, but attendings and residents didn't really answer that they had any training at all during this. We wanted to know what they knew about the pandemic, so we asked about their diagnosis and the methods of transmission. About 74% of participants said that transmission was through droplets, and more than 20% of residents and attendings answered that the virus was aerosolized. We also asked about the diagnostic test. So as we can see, most people answered that the best diagnostic test right now was the swab PCR. And we asked them how where they thought they were more exposed to the virus. And most people answered that it was in closed spaces within the community. Only janitors and staff said that they were mostly in the inpatient section of the hospital where they were more exposed. We asked attendings, residents, and medical students about the treatments that were available for COVID and which ones they thought were more appropriate. So the most appropriate answer, the answer we most see was oxygen and intubation, but there were other answers. Some answered other antivirals like remdesivir. We also had answers for dexamethasone. Other treatments that were being used, like hydrochloroquine, ivermectin, plasma, were mentioned by less than 10% of those groups. We asked them, because in Mexico we had a large number of infections, some people were complaining about the availability of PPE in their hospitals. So we asked them if they thought that they had enough PPE available. And we can see that most people in the survey answered that there was enough PPE. 76% answered that there was enough. But if we see the spread, attendings and residents were the ones that complained the most of the unavailability of PPE in their spaces. We also wanted to see if they had been infected previously. And we can see that even though 72% of the answers were no, I haven't been infected previously, residents and attendants mostly answered that they did answer that they had been infected. So 50 attendings out of 150 said that they had been infected before, and 52 residents out of 100. So those were the most important groups for both. What we concluded for this was that healthcare workers in Mexico are mostly aware of the transmission and the preventive methods that are needed to stop the spread. So that's very important. But we do have to work on using trustworthy sources, because even when attendings and residents use mostly peer-reviewed publications or new publications, 20% cited social media and news outlets as their main source of information, and only 17% have received training. So we need to work on spreading that training and making this this peer-reviewed publications available to other people so that they can use them as sources of information. We also saw that most healthcare workers perceived that we had enough PPE available for everybody. So that was good, but attendings and residents did suffer from shortages. And we need to address these areas to improve survival and reduce local transmission, especially with healthcare workers. Thank you.
Thank you. Very interesting. Look forward to the discussion later. Next, our last presentation, and then we'll open it up for discussion. Feel free to put things into chat, as at least one person has. Andrew Heim, if you want to start sharing your slides. And he's the lead project manager at University of Kansas Medical Center, and he's going to be talking about the TAME study and COVID-19 challenges, how we adapted to the global coronavirus pandemic.
All right, great. Can everybody see and hear me okay?
Yeah, are you in presentation mode?
Let me check here.
Because we're seeing your next slide. There you go. Good.
All right, give me just a second here. Does that look okay?
Now it does.
Okay, great, thanks. Okay, so thank you everyone for joining, and thank you, Dr. Larson, for that introduction. I'm Andrew Heim. I'm a project manager at the University of Kansas Medical Center, and I'm joined by the PI of the study, Dr. Barron, as well. As you can see, Dr. Barron's at the University of Missouri. He relocated there about 7 months ago. So the study did start at the University of Kansas Medical Center, but he kept me on as the project manager when he moved. So I'll be presenting on the TAME study. This is a little bit different than the presentations we just saw. This is a rare disease randomized controlled trial, and it's kind of a success story. One of the few that we've had, I think, where we were actually able to enroll successfully despite the pandemic. So the official title of the TAME study is Therapy in Amyotrophic Lateral Sclerosis, or ALS, best known as Lou Gehrig's disease. So that's the disease that we'll be presenting on today. Here are disclosures, none for me. You can see Dr. Barron's there. And the funding for this study is from the FDA Orphan Products Division. It's an R01 grant 3937. So a little bit of background on this study. It's a phase 2B multi-centered randomized controlled trial, and it's evaluating the safety and efficacy of memantine 20 milligrams twice a day in patients with ALS. We do have a total of 90 participants enrolled in this study that were recruited from 13 centers across the United States. And it does have a 2-to-1 randomization. The aims for this study, aim 1 is to evaluate the safety of a high dose of memantine in ALS patients. The reason why we say high dose there is because memantine is an indication approved in Alzheimer's disease. The standard dose for that population is 10 milligrams twice a day. So we're doubling that dose, which is why we mention high dose there. Aim 2 is to determine if levels of changes in specific biomarkers can correlate to disease progression. Those biomarkers are being processed at Barrow Neurological Institute by Dr. Robert Bowser. Aim 3 to validate the data of the pilot clinical trial, which suggests that there was a 40% decrease in disease progression of ALS using the ALS-FRSR. The pilot trial was done by Dr. Todd Levine, who's a co-PI on this study. And the ALS-FRS is a functional rating scale, and that's widely used as the primary outcome in ALS studies. And then for aim 4 to demonstrate if memantine can improve neuropsychiatric changes seen in some patients with ALS. On the next slide, I just wanted to quickly highlight the sites that participated in this study. You can see we have a pretty wide distribution of sites across the US. We do have 2 in Kansas. We have our satellite site in Wichita participating, 2 in Missouri with Dr. Barron there, and then site 9 is Springfield, Missouri. And we do have Texas and 2 in Texas as well. So how we were able to adapt to the study. Once the coronavirus pandemic hit, what we immediately did was call kind of an emergency meeting with all the study sites, with their PIs, their study coordinators, to get feedback from them and see what they were allowed and not allowed to do at their sites. So, you know, were their sites shut down? Was research staff allowed to be on site? Were they able to enroll new subjects? Could they perform pulmonary function tests, which was a big thing for us? The forced vital capacity and the original set of inclusion and exclusion criteria was part of that entry criteria. So we had to figure out, if we're not able to perform PFTs at the sites, how are we supposed to enroll patients? So we had to kind of think outside of the box there. So once we got all the information and data from the sites, we took all of that to the steering committee and we put our minds together and formulated a modified protocol that was adapted to fit everybody's needs. So what we did is we created an alternate set of inclusion and exclusion criteria that allowed for subjects to be enrolled partially or completely remotely. We were able to use data from their clinic visits within the past 3 months, such as physical exams or for the forced vital capacity that I mentioned earlier. Once we did that, we got it approved by the central IRB and then all the applicable site IRBs. And of course, we sent that to the FDA and to regulatory authorities for approval. Luckily, memantine is encapsulated and can be kept at room temperature, so we were able to ship the medication to the subject's home so that we could do the study remotely. Prior to the pandemic and the altered protocol, the study included 5 in-person visits and 8 telephone visits or remote visits. So the study was kind of already set up a little bit to be done remotely. So we did allow those 5 in-person visits to be done remotely as well if needed. All right, and then this shows kind of an enrollment timeline of how the study looked pre-pandemic and post-pandemic. I wanted to quickly highlight that the national shutdown was right around March 18th, and we got our new protocol approved by April 7th. So that is a pretty quick turnaround time there, as you can see. And then when you look at the timeline here, you can see that enrollment actually increased after the pandemic. You can see subjects enrolled in March was 5, and then 8, 9, and 11 post-pandemic, and that was due to the ability of being able to enroll remotely. So when you look at this, over half of the subjects, 47, in fact, were enrolled in the 7 months post-pandemic, and 43 were enrolled over 16 months prior to the pandemic. Part of that is due to site activation as well. Obviously, when you look at the start of the timeline here, the end of 2018 and start of 2019, that was mostly when just KUMC was activated and maybe one other site. So obviously site activation as we went along played a factor into this as well. But without the remote enrollment, I don't think we would have been able to finish this study on time like we had planned. So in conclusion, we were able to adapt quickly and stay on target for completing enrollment. Kind of the unsung hero of our success story is the IRBs and how flexible and accepting they were to the changes that we proposed to the protocol amidst the pandemic. And then similar studies could be done primarily remotely in the future in rare disease or even common disease. And that's everything. Thank you.